this session by Maël Plessis and Laure Oppenau about 15 puzzles. So with the microphone, it's good to or hold it like that or stick it here to be able to be well listened. And I will let you start. So, sorry, it's not the first one.
Il y a un bouton on-off sur le côté.
Hi everyone. So we are two students from the Institute of Mathematics of Orsay, and we are here to present our project on the game of Taquin, which is also called the sliding puzzle. This game is composed of moving tiles that can only move when they are right next to the empty square, and they can move to the empty square. The goal of the game is to reorganize the tiles in the right order, often to recreate a picture. For our research, we decided to name the tiles with numbers from 1 to 15 to make it easier. So the first problem we had to solve is called the Symboloid problem, and the question is whether or not we can exchange the tiles 15 and 14 without moving the rest of the puzzle. Then we wanted to know if we could make every permutation possible and to determine what permutation was possible or not. So we used this Taquin, but also other shapes of Taquin that you can see over there.
First, we wanted to know find some properties of our puzzles. So our first theorem concerns the number of swaps to reach a position. We introduced the concept of permutation. A permutation is a change in the order of the tiles, and a permutation is achieved by a series of swaps between any tiles. An even permutation is a permutation which is achieved with an even number of exchanges. So, because of a situation, the empty square has to return to its initial position. And because the puzzle is built on a grid, the empty square makes as many moves up as down, and as many moves to the right as to the left. And so the number of exchanges is even. We can say that all the permutations allowed by the rules are even. So with the theorem, we can conclude on the Symboloid problem. Here there is only one exchange, so this position can only be reached by an uneven, not even permutation, and so it is an impossible situation.
So then, to know whether or not we could reach every even permutation, we decided to divide our Taquin in zones with often common squares. And the idea was then to build algorithms to reach the position we wanted to reach.
All our algorithms are built the same way. Let's say those 5 red tiles are well positioned. and we want to place the blue tiles next to the 5 red. So here the 2x2 zone allows us to isolate the blue tiles and then move the red one next to it.
So by this algorithm, we proved that we could make every even permutation in this case, and we can generalize that algorithm to every tachon with 2 zones with exactly 2 common squares. This means that our first tachon is can make every even permutation, since here we can make 2 zones with exactly 2 common squares. It isn't the case with every tachon. This one, for example, cannot achieve every even permutation because there is only 1 common square. So once a tile is in one zone, it can never go to the other one. Thank you for listening.
Thank you very much for your presentation. So now we're going to listen to Arthur Muley about development of innovative reactor models for fuel cycle physics, cause and assessment of their contribution to a transuranic element management strategy. Just to remember for the people that arrived later, I will do this sign when there is just 1 minute left for the speakers to know where they are for the time.
So, hello everyone, and I would like today to introduce to you to my subject research, which is titled, as you can see, Developing Innovative Reactor Models for Cycle Physics Codes and Evaluating their Contribution to a Transuranium Elements Management Strategy. Woo! So, behind this odd words, what I would like you to understand is that by transuranium elements, we understand atoms that are heavier than uranium, and mostly we are talking about plutonium. So, what I want you to understand is the current context. In the French nuclear fleet, what do we have? We have pressurized water reactors. They are 100% all pressurized water reactors, PWRs, as you can see. And those PWRs all use UOX, uranium oxide, which is, just to understand, is enriched uranium. So, those PWRs, by using this UOX, they all are open cycle, which is to say it isn't a cycle at all. We do a little bit of recycling with this spent UOX, which is called MOX, mixed oxide, or just a mix between uranium and plutonium, and it is about 12.5% of the assemblies of nuclear fuel in France. And once we have used them, we store them. We store them away in cooling pools. So, what is the thing to understand and to think about is that at the moment, technologically, we do not have the possibility to recycle everything. Here we go. So, there is a need for innovation in recycling the spent MOX, recycling plutonium and other transuranium elements. And the French government has decided to tackle on this need for innovation with startups and with the France 2030 call to projects. There's different startups, all are giving new technologies, new reactor technologies to tackle on this problem. And we can group them by type of technology. There is 4 new types of technology, plus 1 startup that is always on the PWR, pressurized water reactors, which will serve for my research purposes as a comparison element. Here are the new 4 technologies. And then, what are my goals? The goal is to understand the new physics behind these new nuclear reactors. What I want you to look at is this graph. For comparison, you have the neutron flux spectrum. Behind this complicated words, you just have to understand that it is a good representation of how does a nuclear reactor work. In purple, you have the PWR, pressurized water reactors, that we do know a lot about, and you can see that these new reactors are here. And just by a rule of thumb, you can understand that they are completely different in terms of physics. They are called fast neutron reactors. Why? Because the neutron activity is a lot higher in energy and so in speed than the current fleet of nuclear reactors. So I have analyzed this new physics with a simulation. And there is a first type of result we can draw conclusions from, and that is the required percent of uranium-245, which is to say the enrichment of uranium necessary, and the required percent of plutonium for the reactor to work. And with that, we have different perspectives. We can answer, for example, there's three really important questions from the point of view of the industry. First, is it below 20%? Is the required uranium below 20%? Why? Because if it's any higher, it's considered as military applications, and you do not want your reactor to be considered as a bomb. Then you have the percentage of plutonium. It's good that we have required plutonium, and it's good if it's high, because we can recycle more, but if it's higher than 30%, we cannot do it. Technologically, we cannot do it. So, what are the next steps of this research? The next step would be to create a more complex simulation of these reactors, because right now it is really simplified, and produce data that can be used in another simulation code to evaluate the impact of these new reactors on the French nuclear cycle and on the cycle of the nuclear fuel in France. I thank you for your attention.
Thank you very much for your presentation. So now we're going to listen to Daphne sorry if I say your name. Daphne Osguven and Zhuji Wu. They will speak about the study of the learning task space induced by electronic health record data.
Good evening, professors and students. We're really honored to be here to share our work entitled Study of the Learning Task Space Induced by Electronic Health Record Data for its prediction of sepsis, which is also a part of internship at the EBISC laboratory under the supervision of Ms. Hamidi, and she's my colleague, Daphne, who will present you today. Okay. Oh, sorry. Let's start with sepsis. So what is sepsis? According to the consensus definition, sepsis is a life-threatening organ dysfunction caused by a dysregulated host response to infection. And the body's reaction will cause damage to its own tissues and organs, which may lead to shock, multi-organ failure, and even death if not recognized early and treated promptly. And is one of the most frequent causes of death worldwide, affecting nearly 15 million people per year, and responsible for 20% of all global deaths. And the early detection of sepsis can decrease the mortality rates greatly, which makes the prediction of sepsis onset all the more important. And deep learning is a promising approach for early prediction of sepsis. So to know how it works, let's go through a simple example. Here's a case of the image classification, and here's the data we can input into the learning model, The data is various images of like tigers, dogs, and cats. And it represents the labeled data that the model will learn from. And in the middle is the deep learning model, usually a neural network, and where multiple layers will perform computations on the data and to recognize the patterns associated with each each animal class. And it illustrates what we call a single task setting, meaning the model is designed to perform one specific task. And in this case, identified the animal appears in the image. So for the prediction of sepsis, the idea is quite similar, with the main difference that the idea that we are predicting whether the patient will experience sepsis instead of cats and dogs, and using the medical data instead of just simple images. And the data are obtained through different modalities, such as lab measurement and vital signs and clinical time series data. And the data is taken from hospital in the form of electronic health records, and a deep learning model will be trained on this to recognize the patterns associated with this sepsis.
So the database we're using is called Mimic4. It's a large database of the identified patients admitted to emergency department or the intensive care unit of a medical center in Boston. It is a very famous, complete, and challenging database consisting of 65,000 patients admitted to ICU and 200,000 patients admitted to the emergency department between the years 2008 and 2022. So together we've seen that single task setting is limited when predicting sepsis. Sepsis prediction is a very complex challenge, so it needs more featured methods. And various approaches have been proposed in the literature, including multitask learning. The difference from single task setting is that in multitask learning, we train a single model to learn from different tasks simultaneously. The main advantage that it can leverage knowledge sharing across tasks and deal with limited amount of data, which is the case of health applications. And in the first example, we see that the authors have chosen to learn from two tasks simultaneously, the sepsis onset and remaining time before sepsis onset. In another example, the authors have chosen relatively similar tasks, but they're still distinct. Task formulation is an essential challenge of these models, and it can influence the final model performance a lot. And this is a question we're asking in our research. And in another example, Lin and al. Show that different combinations of tasks result in different model performances, and some result in better performances. Specifically, combinations of tasks with a high inter-task relationship lead to better performances. This is another question we're asking in our research. So overall, using the Mimic4 database for training our model, we're aiming to investigate the impact of task modeling on the model's performance in the context of sepsis prediction and improving knowledge sharing across tasks using these insights to build better predictive models. Thank you for listening.
Thank you very much for your presentation. So now we're going to listen to Maxime Pilorget, Lucien Vieillard-Baron et Vianney Garnois. And their presentation is about a disaggregated view on technological change.
Okay, so hello everyone. Today we will interest ourselves in the technological change problem. So the main questions are how to make growth more inclusive, which quantity of the growth is necessary to fight climate change, but also what leads firms to automate their production. So those are all questions which are linked to the technological change, which is a central problem in growth economy. The main question of our presentation will be, will the establishment of a carbon tax lead firms to produce more by still emitting the same quantity of CO2? So another way to say that is if a rise of the price of energy will lead firms to innovate in the direction of energy. The whole project is based on this equation, which is a Leontief production function, which is a famous production function in economics. And there you have the production of a sector of the economy, for example, the number of cars which is produced by the car sector, but also some inputs, which are capital labor, which are used by this sector. And the main parameter, so the one that interests us in this project, is the technologies, the technological parameters, which we will try to estimate.
So in economics, you can conduct experiments. So we had to work on real economical data. So we work on a claims database, which is for capital, labor, energy, materials, and services. These are projections data on 30 years, and it is separated within 50 sectors, and in every sector we have the data for the inputs that are used to produce in this sector. So the five inputs that are the claims, and we have also the production data, which are the quantity and the price of outputs that we produce.
So, in order to compute our model, we need to determine the level of technology of a resource. So, we are going to try two main ideas. The first idea is that, for instance, if the price of energy is increasing, a company will be more likely to invest money on the development of more energy efficient machines in order to tackle this price evolution. We are also going to try another idea, which is that we have an effect of induced innovation, which consists of the increasing of the price of the energy will provoke the increase of the level of technology of other resources, such as materials or services. So in order to do that, we are going to work with a temporal series consisting of the prices of the different resources and also the level of technology. And what we are going to do is making like auto-regressive variables in order to determine causality and also correlation between the prices and the level of technology. So the goal is to determine if a structural shock of the prices will have an effect or not on the level of technology of resources.
So we conducted our analysis on 56 sectors times 5 combinations of inputs, and the main results are here. So first, the intuitive theory is confirmed. Secondly, we observe in a lot of sectors induced induced innovation effects. And to look more on the energy side, we see that on the lot of key sectors, however, we don't have an innovation of technology on energy linked to the rise of the price of energy.
So, as a conclusion, the establishment of a carbon tax leads to small innovation in the direction of energy, but so it can lead to decarbonization, but not necessarily to degrowth.
Thank you very much for your presentation. Now we're gonna look to a video of Yona Kouam about the modeling and improvement in cochlear implants.
Hi everyone, my name is Yona. Hi everyone, my name is Joanna, and today I will present my summer internship modeling and improvement in cochlear implant, which is provided by Professor Shakir and Dr. Edlin. So you may wonder, why optimize cochlear implants? And that's a good question. They are the most widely used prostheses in the world. These devices enable thousands of profoundly deaf individuals to regain near normal hearing and lead a normal social life. However, they are still challenging to overcome. By profoundly deaf, it means that the signal are damaged or non-existent, and therefore the signal is not transmitted. So one of the main challenges is to optimize the diffusion of electrical currents in the cochlea. The cochlea is that spiral-shaped organ located in the inner ear, responsible for converting sound vibration into electrical signals that the brain then interprets as sound. In theory, you would expect one electrode to activate one bundle of nerve fibers. As you can see here, no electrode, no fibers, where in reality, one electrode can activate an overlapping population of nerve fibers, thereby reducing the precision of the stimulation. To overcome this problem, it is crucial to understand how different forms of electrical pulses activate auditory nerve fibers. For several years, strategies have been tested to limit this diffusion, particularly by modifying the shape of electrical pulses. Most implants use biphasic, symmetrical, and rectangular pulses. However, recent studies suggest that round pulses that you can see here could be more effective in activating the auditory nerve fibers. And so, Jean-Marc Islin's team has conducted extensive research on the effectiveness of round pulses. You can see there the experimental apparatus. They perform electrophysiological recordings in the primary auditory cortex of guinea pig by inserting a matrix of 16 electrodes. Then, they used a bundle of stimulation electrodes that they inserted into the cochlea, and the first electrode of the bundle, the most apical one, was actually used as the stimulation electrode for the auditory nerves. The response evoked by the different pulses were recorded and analyzed. So there you can see the result with the raster plot, which represent the row evoked responses obtained in the auditory cortex. For two simultaneous recordings labeled channels 6 and 7, with increasing levels of charges from 3 to 31 nanocoulomb, and each dot represents the occurrence of an action potential, and also the dashed line indicate here the threshold value obtained for each recording, so the threshold represents the value for which we're sure it's not a spontaneous activity. So the experiment showed that RAM pulses trigger stronger responses in the primary auditory cortex with lower current thresholds, and these plots illustrate the difference in effectiveness between rectangular and RAM pulses. So these results pave the way for new stimulation strategies to improve cochlear implant. So based on the results, we hypothesized that RAM pulses more completely activate the dendrites of neurons, whereas rectangular pulses would only activate the signals, which are actually further from the electrodes. So to try to understand why RAM pulses are more effective, modeling the activation of auditory nerve fibers by different forms of pulses seems necessary at this stage of the research. as it will allow us to better understand the factors that lead to this greater effectiveness of RAM pulses, and this constitutes basically the objective of my internship. So using the neuron stimulation environment, we will model a nerve fiber. This will involve realistically simulating the ionic currents present in the nerve fibers and understanding how these different forms of force influence the activation. By applying currents and analyzing the propagation of the action potential, we'll be able to observe the dispersion and loss of amplitude at different points on the fiber. So this will allow us to better understand the result obtained by Dr. Edlin's team. So in conclusion, the use of RAM pulses seems promising for improving the effectiveness of cochlear implants. And to that, there are two aspects. First of all, RAM pulses offer reduced diffusion, which indicates a better selectivity. And second of all, they reduce energy consumption, which is crucial as constructors' current goals are to have totally subcutaneous implants with long-lasting batteries as to avoid frequent replacement operations. So, by modeling this effect with neurone and comparing the result with experimental data, we can optimize the functioning of these devices and improve the quality of life for cochlear implant users. Now to finish up, you can skim through the references if you are interested. And I want to thank as well my supervisors, Professor Jakir and Dr. Edlin, for their help in our project. And thank you for listening and have a good day.
So now we're going to listen to Anna Dia-James about solar energy storage and the release using acid-triggered molecules.
Hello everyone, my name is Anna Dia-James and I am a Master's of student at University Paris-Saclay. And I'm currently doing my master internship at the PPSM laboratory in ENS Paris-Saclay, where I'm supervised by Remy Métiers, Keitana Dokhtani and Leah Shakur. So today I want to tell you about a very interesting, fascinating idea. So, what if I tell you that using chemistry, you can store the energy from the sun in the molecules and use it when the sun is not shining? I'll tell you exactly how we can do it. Let me begin by telling you about a problem that all of you know. There is a rising demand for energy worldwide, and in fact, this problem is not going to go away anytime soon. In fact, the more comfortable our lives get, the more energy we consume. So we need alternative ways to meet this energy demand. Solar energy is seen as a productive solution to address this challenge. But there's a catch. The solar energy production peaks during the day and the generation and the usage during the day is low. And at night, when we need the energy the most, the generation is very less. So what we need is ways to store energy and use it when we need it later. So now you might ask me, we already know how to do it. There are solar panels which does exactly the same. But there's a problem with this, because the solar energy is converted into electricity and then it's stored in batteries. There's nothing wrong with batteries. Batteries are produced in large scale and it is very effective today. But these batteries are made of rare earth metals. It's not very sustainable after all. So that's where we need a new solution. This is a molecular solution. Let me introduce you to a new concept. Now imagine that you can store your molecules. No, you can generate energy from molecules. That is exactly what molecular solar thermal systems does. There are specially designed molecules which can absorb and store energy, and then they can later release it in the form of heat. The advantage is that there is no electricity involved and the molecules, the energy is stored in organic molecules, plain and simple. Now let me tell you what is it that we can do with most systems. Using most systems, imagine coming home on a cold winter day and at night when you come back, your home is heated up, but not from electricity, but from the energy that is stored in the molecules. And the sun, the energy can be stored from days before. The energy can be from the sun that was shown days before. So this is clearly an advantage because you can heat up homes from the energy that was from the summer or days before. So I've told you now how the system, what you can do with it. Now let me explain the science behind it. So I told you this is a molecular system, so we need molecules. So there are specially designed molecules that exist in two different forms. The molecule in the open form, when you shine light on it, it like flipping a switch, it changes into a closed form, and the energy is stored in the molecule. And then when you add a trigger, the energy is released in the form of heat and the molecule goes back to the open form. And this cycle can be repeated multiple times as long as you need it. So in our group, we study the molecules where acid acts as a trigger. So few drops of acid and the molecule goes back to the open form and releases energy. So to do this, we need molecules. These molecules are synthesized by our collaborator Dr. Yu in ICMO laboratory in Paris-Saclay. So during my internship, I explored a few molecules which are synthesized in ICMO, and we need these molecules to absorb sunlight. So I study their spectroscopic properties. And I told you we study the molecules where acid acts as a trigger. So I studied their acid-base behavior. And most importantly, we need these molecules to store energy. After all, that's what we are studying. So I do some computational calculations to analyze the energy storage capabilities of the molecules. So that brings me to the end of my presentation. So if there is one thing I need you to remember from my presentation is that molecular solar thermal systems most offers a reusable, renewable, and an on-demand solution to address the energy problem. I need you to think of most molecules as batteries, but with molecules. And this is just a small step in the direction of solving the energy problem. I thank the organizers of the Junior Congress for giving me the opportunity, and I thank you for your attention.
Thank you very much for your presentation. Now we're going to listen to Nathan Chevalier, who's going to speak about a very different subject. He's going to speak about geopolitics and trade, an application of the gravity equation.
Okay, so hi everyone, so can you listen to me? So I'm going to present you my second year master's thesis, which has been partly realized at CEPI, which is the research center in international economics of the services of the Prime Minister. So to give a bit of context to my presentation, so as you may know, we observe increasing geopolitical tensions in the world in recent years. So I talk about mainly the conflict in Ukraine, but also in a less violent way about the tensions linking USA and China. These have important implications on international trade, and especially for war, which has a particularly destructive effect. But it's also the case for non-violent physical events, which are not recorded in the previous literature that basically consists in geopolitical risk indices that are computed using only English-speaking sources and do only census physically violent events. So what I do to solve this problem, I use the global database of events, language and tone, which is basically a machine learning algorithm that is going to extract information from articles in 100 different languages. So to give you an idea of the size of this database, it currently weighs around 8 terabytes, and it grows an additional terabyte a year, so because it's updated every 15 minutes. So it needs to be sorted, it needs to be processed, and that's basically what I do to compute a brand new geopolitical events database. And from this I compute two indicators of bilateral relations. The first one is the importance, which is basically, if you read the example here, the share of articles regarding one specific country regarding two countries in the whole amount of articles regarding one specific country. and it's going to quantify the importance of the bilateral relation. And then I compute the shade, so I sort all events extracted from articles in categories, among which declarations, actions, both be they positive or negative, and then violent events. And the shade, for example, for violent events is going to correspond to, for example, the share of articles evoking violence between Ukraine and Russia among all the articles between Ukraine and Russia. And so it's going to describe the nature of the relationship between the two countries. And to estimate the impact of tensions on bilateral trade, I use the gravity equation of trade, which is basically the economic analogy of Newton's gravity equation. It doesn't look that good here, so I will explain it to you using clear examples. So let's take, for example, Russia and Ukraine, which are two rich countries in terms of GDP, that you can see in the equation, and are additionally very close to each other. So the distance term is very low, and so they will trade more, okay? If you take now Australia, Australia is also a rich country, but it's very far from Russia, so they are going to trade less. If you take now Mauritius, Mauritius is a very small island, not very rich, but also very far from Russia, so they are going to trade even less. What I estimate, it's the beta 4 here, is the impact of the tensions term, which is basically a combination of the two indicators I computed previously, and corresponds to the impact of tensions on bilateral trade, so if we add tensions between two countries. So to give you a first taste of answer, since I only got the results from this week, so if you want to ask me, feel free. But I only give some stylized facts. So if I represent the importance left and the shade for violent events right, together with total bilateral trade between Ukraine and Russia, you can see here that when both indicators are high, so when both the importance and the share of violent events between the two countries are high, you experience a very important reduction in bilateral trade. But remember that I told you about non-violent events as well, so if you plot the same but for negative actions, among which you can register tariff increase, for example, by President Trump during the first trade war in 2019, you observe the same thing. So to conclude about this project, I deliver two important contributions, so a new open source geopolitical events database, so that is going to be delivered soon, and new impact estimation, so once again that I got from this week, so if you want to ask me for additional stylized facts or estimations, feel free. And it's going to be continued in a PhD thesis linking the environmental aspect soon. Thank you for listening and feel free to ask.
Thank you very much for your presentation. Now we're going to listen to Elise Miller, who's going to present the impact of microbial metabolites on the developing brain and behavior.
Good evening. My name is Elise Miller, and today I'm going to be presenting research from a 6-month internship in the McAllister Institute on the impact of microbial metabolites on the developing brain and behavior. Oops. So, to begin, the gut microbiota is the collection of microorganisms that inhabit the gut. We know that there exist bidirectional pathways of communication between the microbiota, the gut, and the brain, which we collectively refer to as the microbiota gut-brain axis. So one example of this is during situations of stress, such as before an exam, signals pass from the brain along the vagus nerve to the gut, leading to alterations in gut motility that you might experience as feeling sick to your stomach. Signals can also pass from the gut microbiota to the brain. So in germ-free mice, which are mice that are raised under sterile conditions that have no microbes in their gut, these mice have an altered stress response when compared to mice with a normal microbiota. We also know that across species, alterations to the composition of the normal composition of the gut microbiota lead to a higher incidence of emotional deficits or psychiatric disorders, with these deficits being more severe when this alteration happens at a young age. This is understandable as the early life period is a critical time for the development of both the gut microbiota and the brain. Here you can see a timeline of human brain development aligned with a diagram of the growth and diversification of the gut microbiota. And as you can see, these first 3 years of life, that period from birth to weaning, are a very significant time for the development of both. So my research centers around understanding the role of the microbiota gut-brain axis during early life development. My team specifically works on understanding the role of microbial metabolites in this gut-brain crosstalk. So to understand what these are, let's take a look at 2 bacteria that are some of the first to colonize the gut, E. coli and B. longum. These bacteria are able to break down tryptophan into the molecules indole and ILA, and these are microbial metabolites. Indole and ILA can then pass into the bloodstream and then move from there and cross through the blood-brain barrier into the brain. Extensive work from my lab has shown that heightened indole production increases anxiety-like behavior in mice and rats, and we see that in human subjects, B. longum probiotics significantly reduce anxiety and depression scores, so we can wonder whether this might be through the action of ILA. Our present study seeks to ask on what role does indole and ILA production by the intestinal microbiota play on the host's behavioral development. So to do so, we look at 2 experimental groups of mouse pups that are colonized with just E. coli and B. longum in both cases. So the first group is capable of producing both indole and ILA. In the second group, these bacteria are capable of producing indole, but not ILA. And we then assess cognition, memory, and anxiety-like behavior in these animals. So how do we do this? We begin with germ-free mice that have no microbiota, and we perform all work in these sterile boxes called isolators. We couple mice for a period of 24 hours, which allows us to predict the expected date of birth, and we then introduce the bacteria to the mothers 1 day before birth and shower the mouse pups with the bacteria 1 day after birth. We perform all of our behavioral tests between postnatal days 6 and 44. So what do these tests look like? One example of a test of anxiety-like behavior is the step-down test. So in this test, we place a mouse on an elevated platform, and we measure the amount of time it takes for the mouse to descend from the platform. More time to descend from the platform is considered more anxiety-like behavior. So here on the left, you have the mice that are capable of producing both ILA and indole, and on the right, the male and female mice that have only indole. And so what we see is that the mice with both indole and ILA exhibit more anxiety-like behavior. And so we have shown that ILA seems to increase anxiety-like behavior, but further studies will be needed to assess what differences exist in these brains on a cellular level. And so, thank you for your time.
Thank you very much for your presentation. I'm just opening the door if someone wants to enter. So now we're gonna listen to Noshim Anjum Monisha. Did I say it correctly? Yes. Okay, perfect. So she's gonna present us genetic architecture of resistance to Septoria triticii blocked in traditional Tunisian durum wheat landraces.
So, hello everyone. I don't have to repeat the big title. It's basically my M2 internship topic, which is conducted in INRAE Bioger, and under the supervision of Thierry Merston. And I would also like to thank the people who are involved in this project, Safé, Frederic, and Emma. Durum wheat is one of the most important crop worldwide, and Mediterranean region like Tunisia is the largest producer and consumer. So these Tunisian crop are probably more often in our plate, as this wheat is used to make pasta, couscous, semolina. Is essential for food security. That's why it's very important to protect this crop from the disease. So here we have the map of Tunisia from where each of the land races are collected and we used in our experiment. So what are these land races? So they are very genetically diverse and well adapted to local condition. And they have shown remarkable potential in limiting this STV disease. So what makes them successful is just not having the resistant gene, is how these genes are structured and interact within themselves. So I couple of times mentioned the STV is a disease that is caused by Zymoceptor and here is the whole life cycle of Z2TC. So what happened is that these ascospores coming from the fungus crop debris enter to the leaves through stomata. They grow inside the plant silently without showing any symptoms, but 14 days they cause this primary infection. And then they release the pycnidiospores that infect one leaf to another by contact or the rain dispersal. Since it involves the sexual and asexual reproduction, it evolves quickly and it's hard to maintain this resistance. So to answer these questions, we aim to study how this identify the genetic architecture of these three land races. Then we also aim to understand how this gene respond to different isolates of Z3TC, and later with this information, we propose new strategies for deploying resistant elite varieties. So here is our pathology. So basically these seeds are coming from Tunisian gene bank, which are like self-population after 2 generations. They were sown and grow for 3 weeks, and then we start our pathway assay. What we did here is that we cultured the Z3TC and then make the suspension with the water and then carefully inoculated on the leaf surface with a paintbrush. And then we observe the symptoms in day 14, 20, and 26. So on our phenotypic side, we have the max sporulation, area under disease progression, necrotic, and sporulation as well. So here, what is the pycnidia is that the black spots, and the necrotic is the brown and this dead area. And on our genotyping side, it is generated from the SNP array. And what is SNP or single nucleotide polymorphism is just a tiny spelling difference in DNA that makes us different from each other. It tracks the genetic information. So with this, we perform the genome-wide association study, combining this phenotype and genotype to understand how this genetic marker is linked to the phenotyping trait, in our case, resistance to STV. So this GWAS is like a genome-wide association study too, where they often import as a Manhattan plot. So here the different color represents the chromosomes, and this blue line is the threshold level. Each dot represents the genetic marker, and in the y-axis is the minus log 10 p-value. So higher the dot is, higher the resolution. In our case is chromosome 6, 11, and 17. So what it tells us that it narrowed down the candidate gene, which needs to be further validated by the functional studies. So to conclude, it just not only tells us how they are genetically diverse, it also helps us to efficiently limit the STB epidemics. And in a bigger picture, it contributes to stable yields and sustainable agriculture. Thank you so much for your attention.
Thank you very much for your presentation. Can you leave the door open, please? So now we're going to listen to Charline Coudry. She's going to present us Neutrality in Transition, Legal Challenges in Outer Space and Cyberspace.
All good. Well, good afternoon, everyone. I'm happy to present this topic, which is my research thesis that is conducted as part of the PhD track program, and that will be continued hopefully next year in PhD in international law. So it has to do with the international law of neutrality. And this is an important question because the war in Ukraine has raised several questions in international law about what it means to be a neutral state today. So we have had an attack, a cyber attack on Viasat infrastructure. We have OneWeb and Starlink providing connectivity to troops in Ukraine. We have GNSS use, GNSS being GPS, Galileo systems to guide missiles, and we have also the provision of satellite imagery to Ukraine. So what does it mean to be neutral today, and can we use a 1907 framework, a 1907 convention to understand what it means to be neutral today? So what I am doing is interpreting international law. And to understand what I'm doing, it's important to understand the two main conventions on the law of neutrality. So in 1907, states have gathered to codify two important conventions about the law of neutrality. The Convention on the rights and duty of neutral power in case of war on land, and the rights and duties of neutral powers in case of naval war. And so can we still use them today? Well, to answer to that question, it's important that you understand what an international lawyer do. So we do not always stick to conventions because we have two articles saying that international law is broader than just treaties. So first we have Article 38 of the ICJ Statute, which is the International Court of Justice, that considers as sources of law not only treaties, but international custom, which is the practice of states that states consider as binding as law, and also judicial decisions and the teachings as of the most important professors of international law as a way to determine the rules of law. We also have the Vienna Convention on the Law of Treaty, which helps us to interpret treaties, and this article also mentions the recourse to supplementary means of interpretation, including preparatory works of the treaties, or, as I mentioned earlier, the writings of professors. So regarding the law of neutrality, I first have to interpret the treaty in light of its context, so the travaux préparatoires. And regarding cyberspace and outer space, this is quite interesting because, especially for cyberspace, we see that in 1907 there was some consideration about telegraphs, and so we can apply some of the articles of the convention in today's world thanks to these provisions on not electronic communication at the time, but telegraphy. We also have a really interesting International Court of Justice case, not a case actually, but an advisory opinion, which states that the principle of neutrality are customary international law. And as I mentioned earlier, this is a source of international law, which means that the law of neutrality is not just about the convention, but also about principles in customary international law that, really interestingly, can apply to all international armed conflict, whatever types of weapons might be used, including cyber weapons and weapons in outer space. And so we have really interesting manuals, such as the Tallinn Manual 2.0 or the Oslo manuals, written by professors who have been focusing on this idea, and specifically for outer space. The Oslo manual says that neutral states must not knowingly allow space systems and assets to be used by belligerent states in the armed conflict. And so all of this is being treated in state practice because states are beginning to include in their national positions the law of neutrality for their cyberspace and outer space operation. And I would like to conclude this presentation saying that we have a lot to study on the law of neutrality applied to cyberspace and outer space. But state practice is confirming the fact that there is room for the renewal of the study of neutrality law. And I would like just to mention, I don't know if there are any Danish people in the room, but Denmark has included the fact that the law of neutrality extends to outer space in their national position. Thank you very much for your attention.
Thank you for this presentation. So now we're going to listen to Berliana Novianita, who's going to present ultrasound-stimulated polyisobutyl cyanoacrylate-based polymer microbubbles for drug delivery. Sorry if I said missaid the polymer. So.
So, hi everyone, I'm Berliana and today I'm honored to join you to present my work on ultrasound-stimulated polymer microbubbles for drug delivery, more simple. And this is my work, my Master 2 internship at Laboratory for Classical Translational Science. I'm supervised by Cedric Soviet. So, I will start with the question, what are microbubbles? Founded in 1960s, when this guy accidentally created air bubbles during a rapid injection of contrast media during heart scanning experiments. It turned out that actually not only the contrast agent that helped to increase the contrast, but also these air bubbles because its ability to reflect the ultrasound waves. So since that time, people are intentionally shaking the contrast media solution to create air bubbles to increase the photo imaging. But the problem is the size are not very well defined, and these bubbles are not stable in blood circulation. So that's why for many decades we are trying to develop more microbubbles with more stable properties and also with different gases and different composition of shell. And even today, researchers are trying to create even target-specific microbubbles, which it can targeting specifically to sites in our body by adding conjugate adhesion. So in today's definition, microbubbles are gas bubble with a size of 1 to 10 microns with stabilization with shell. Even though originally microbubbles are used for ultrasound contrast agent for medical imaging, but now it's also have gained interest as ultrasound-mediated drug delivery system. So maybe you're questioning why microbubbles are that interesting for drug delivery studies, because we already have nanoparticles, for example. Let me show you. So imagine we will deliver drug assisted by microbubbles. So either we can co-inject drug plus microbubbles, or we can load the drug on the microbubbles, or even more sophisticated, we can use functionalized microbubbles that can target directly to the site. And upon delivery, we will employ ultrasound that will oscillate the microbubbles. And actually, this oscillation can have two effects. First, of course, to facilitate the drug release. And secondly, which is very cool, this oscillation can enhance the membrane permeability, by creating temporary pores, and it will allow more drug passing through. And this phenomenon called sonoporation. So, jumping into our study, which is we are trying to develop polymeric microbubble with isobutyl cyanoacrylate with a cool method found invented by our former PhD student, which is acoustic cavitation, which is changing mechanical agitation with ultrasound to make gas bubble formation. And during the synthesis, to acidic water, we will add surfactant, which is going to be the bubble template, for the cyanoacrylate to polymerize and create shell around the bubble gas. So in this study, we have three main experiments. Firstly, we will try to incorporate different gases And secondly, we try to optimize the drug loading. And finally, we try also to lyophilize microbubble suspension into powder in hope that in clinical setting, we can just directly resuspend this powder with drug solution. So in the first experiment, we are very happy that we are succeed to produce microbubbles with different gases with a mean size around 3 microns and no size is greater than 10 microns, which is very important because if it's too big, it's going to blockage the blood vessels. And also we have negative values of zeta potential, which is beneficial to prevent the aggregation during the storage. And also we can produce at least 10⁸ microbubbles per milliliters, which is very sufficient for clinical testing, for example. Secondly, we try to use a dry model, which is Gado-Boutro, but it's very challenging because cyanoacrylate is hydrophobic, so it's hard to load. So our idea is just to try to increase the hydrophilicity by adding polysaccharide, and as a result, we see a slight increase in the loading capacity of our microbubbles. And lastly, we try to lyophilize with 2 different concentrations of sucrose, and we see that actually 5% is enough to protect the spherical structure of microbubbles due to the damage of the freezing and, well, some shrinkage in the oxygen and nitrogen microbubbles. And so what next? So thanks to our cooperation with iBrain and Université de Tours, we will continue to learn the acoustic response of microbubbles to the ultrasound, and eventually we will do in vivo sonoporation study for drug delivery to the inner ear in a shape model. So in this moment, I would like to thank my supervisor and also all laboratory members and all of you for your attention. Thank you.
Thank you very much for your presentation. So now we're going to listen to Timothée Petitjean. Petit-Jean, oui. Okay, Petit-Jean. About the prediction of chemical reaction properties, a journey between chemistry and computer science.
Hello. Are you awake? That's the end of the day. Okay, thank you. So today I want to talk a bit about what we can predict about chemical reaction properties using chemistry and computer science. But to start, I want to ask you a few questions. Do you know what this is? And this? And this? Those are all technological revolutions which changed our way to live. Can one imagine a world without our phones and computer? I mean, half of you are working right now on your computers. Well, it's the same in science. Technological advancements always serve to drive further science. Now, what's the latest technological revolution you've heard of? You don't know ChatGPT? You know this generative artificial intelligence platform. This is based in machine learning. So some talks have presented machine learning before, but not in chemistry. However, in chemistry, it's a really hot and recent topic. For example, it received in 2024 the Nobel Prize in Chemistry, so it's really recent. But how does it work? What's machine learning really? What the main task of machine learning is, for example, to predict things. For example, I want to predict that this is a banana. How does it work? I give to a machine learning model a training set composed of fruits, for example, banana and apple. I give it to a model, and hopefully it will be able to do accurate predictions about which fruit is a banana, which fruit is an apple. Now, how can it be of use in chemistry? Let's take an example. That's vitamin B12. Do you know how much time it took to synthesize this? 12 years, 100 people. Why so? Because to synthesize this very complex molecule, you have many individual steps. So you go from compounds to intermediates, but this doesn't look as our product, right? It's because you have many intermediates before arriving to this final compound. And for each of these single steps, you can vary the conditions. For example, you can vary the temperature, the pressure, the solvent, everything, the concentration, etc. And the issue is, if a step doesn't work, then you have to imagine another pathway or another pathway. That's very complex, right? And this is because every reaction, we do them in single batch. You do one reaction at a time. And one idea to prevent this, so to accelerate further the development of synthesis, is to do many reactions at the same time. If we do 3,000 reactions, we go faster than if we do a single reaction, right? So that's called high-throughput experiment. And another idea is, what if you could actually predict that the yellow path is the one that is going to work? Well, doesn't that look something I talked about before? Machine learning, of course. Maybe we can predict that the yellow path is going to work with machine learning. And that's basically what I want to do for my internship. So it's like combining high-throughput experiments to generate big data sets and machine learning to have fast optimization of reaction. Now, how does it work in chemistry? That's different, because the model doesn't understand molecules, right? I mean, it's just a drawing. So what we have to do is we have to transform this molecule into a figure, into a vector which is a suite of figures, and then we will be able to give it to the model. How do we transform this molecule into a vector? There is several ways. One way is what we call a fingerprint. We all have fingerprints, right? So it's just a pattern in things. For example, in an apple you have seeds or leaves. In banana you have a fixed skin and it's yellow. And we can do exactly the same with molecules. So we have an oxygen or it's an acid. The issue is it only contains structural information. So what we can do is, it's only structural information. So what we can do is maybe give more informative information, for example, the mass of the fruit, the color or the length. Well, we can do this in chemistry using what we call, as chemists, DFT. It's a bit complex, but simply it just gives us, for example, the energy of the molecule, the mass of the molecule, etc. Once we have done this, it's great, right? Now we can apply this to, for example, single reactions. So this is a carbon dioxide incorporation to unite this black and orange moiety. And then we do high-throughput experiment to generate many reactions. We give those reactions to a machine learning model, and hopefully we'll be able to do accurate predictions on the reaction success. So hopefully with this, we'll be able to unite chemistry and computer science to accelerate further development of reactions in chemistry. Thanks for your attention.
Thank you very much for your presentation. So now we're going to listen to Margot Ginest. She's going to present us the underlying occult themes in Margaret Cavendish's materialist philosophy.
Good afternoon, everyone. I'm Margot Ginest, a student at the Department of English at the Paris-Saclay. And I'm going to talk to you about how in 17th century Europe, a redefinition of science, commonly known as the scientific revolution, occurred. With it, magical interpretation of natural phenomena seemed to have been discarded as unreliable and unscientific, in theory at least. Fantastical creatures and magic in general were no longer sufficient causes to explain natural effects. However, reading the text from the epoch, it seems that magical and esoteric worldviews still pervaded the scientific discourse at the time. For my master's dissertation, I chose to study the works of that epitomizes the in-between stages of magical, fantastical, and supernatural explanations in the 17th century by studying the works of Margaret Cavendish, an aristocrat and natural philosopher from England. She defended a profoundly materialist vision of nature, arguing that the latter was infinite, self-moving, and living. This representation of nature, which one can call a vitalist materialism, was opposed to a mechanistic vision of nature in which all parts are just parts of a machine, nature, and moved by an exterior force. Cavendish strongly opposed the idea of an external force moving nature about, arguing on the contrary that all parts of nature have movement, sense, and reason. Her theory of nature can therefore be called a holozoism, the endowing of matter with life, and a panpsychism, in that she conceives that all constituents of nature as having sense and reason. As a materialist, Cavendish argued that the role of natural philosopher was not to elucidate the mysteries of the immaterial and the supernatural, as nothing in nature can be called immaterial. Natural philosophers study nature, therefore all things material. This leads Cavendish to adopt a skeptic stance, considering supernatural phenomena, arguing against the existence of demonic witches, for instance, or against the search of the philosopher's stone by alchemists. However, her philosophy and her poetry are imbued with references to the supernatural, such as she mentions creatures such as fairies, for instance, and her representation of nature bears much resemblance to that of alchemists and other hermeticists of the epoch. Natural elements are governed by relations of antipathies and sympathies. She posits a cosmic harmony at the heart of her cosmogony, and there are correspondences between a microcosm and a macrocosm in the world. Well, these are all elements that are defining features of Hermetic theories in early modern texts. While imbued with the Hermetic representation of nature in the early modern period, she nonetheless criticizes the belief in magic and in the immaterial. She always borrows from supernatural theories or Hermetic theories, but incorporates what she borrows to her philosophy, which is material. Therefore, sympathies and antipathies, for example, can exist if they are material. So she replaces all these references to within a natural order where everything is self-moving and material. Studying the extent to which the natural philosophy of one of the staunchest defenders of materialism at the time truly shows how pervasive some esoteric explanation of nature and its phenomena were at the time of the scientific revolution. In particular, the dialogues between Cavendish and other famous philosophers of the time, such as René Descartes, Henry Moore, Joseph Glanville, Jean-Baptiste van Helmont, or Thomas Hobbes, show that hermetic and magical representations of nature and the universe were still widespread at the time, and not just in Cavendish's philosophy. It thus appears that all the previous explanations for natural phenomena were not swiped away by the scientific revolution. We can argue that the scientific revolution was an age where magic and esotericism were still defining features of many theories of nature. Thank you very much for your attention.
Thank you very much for your presentation. So we're going to listen to the last presentation. The presentation of Nelly Day. Nelly Day, who's going to present biophysical characterization of hydrophilic domain HD of H-SOF2.
My name is Nelly, and today I will tell you a little bit more about my master internship project called Biophysical Characterization of Hydrophilic Domain of HO2 enzyme. First of all, why do we need to study HSOF2? It stands for human endosulfatase 2, because its normal function is to support tissue communication and development. It regulates growth factors and morphogens. And when it's dysregulated, it causes cancer metastasis and angiogenesis. It's already been proved for several cancers. On the level of its enzymatic activity, what it does, so on the cell surface of our cells, we have a molecule called heparan sulfate proteoglycan. It consists of the core protein and long chains of sugar molecules, polymers. If we zoom in, they can be sulfated at several positions. And H-sulf removes at a very precise position this sulfation. And this process is called 6-O desulfation. But even though we know its enzymatic activity, the high-resolution 3D structure is not yet resolved, mainly because of the part of the molecules called hydrophilic domain. So the whole protein exists only as alpha-fold 2 prediction. What do we know about this domain is that it has high affinity for substrate, it has no sequence homology, it has potential multimerization element, it has also intrinsically disordered regions that do not adopt any secondary structure, and it makes the whole enzyme resistant to X-ray or NMR studies. So the goal of my internship is to understand its structure, folding, and interaction separately from the whole enzyme, with a technique called OH radical footprinting mass spectrometry. It's basically, you create OH radicals from hydrogen peroxide, then you irradiate your protein with them, and OH radicals primarily modify exposed amino acid side chains. Usually this is done at synchrotron, such as Soleil, but with a new development machine from Genex Technologies, it's possible to do it on the bench. So it's easy, it's automated, and doesn't require any expensive or complex machines. Then the protein is enzymatically digested to our smaller pieces, peptides, and subjected to LC-MS analysis, also called bottom-up proteomics, which consists from nanoliquid chromatography separation of those peptides and their detection with mass spectrometry. From the liquid chromatography part, we can quantify oxidized peptides. And from the mass spectrometry side, we can identify precisely the oxidation sites, because OH radicals modify amino acids by already known mass. Taking together all the results, we can measure the protein topography, also you can say solvent accessible sites at peptide or amino acid resolution, which of course requires you to have high resolution mass spectrometry. And we can understand which amino acids are more at the surface and which are more protected. But we of course need to compare that with AlphaFold predictions. So in conclusion, this technique is a complementary structural technique to map topography, especially for proteins that resist commonly used methods such as X-ray or NMR, which is ideal for our protein. And knowing hydrophilic domain structural aspects helps to us to understand 3D organization of the whole enzyme. And it's very useful for potential specific HSOF2 therapeutic targeting because it's involved in cancer. And thank you for your attention.
Thank you for your presentation and congratulations to all the presentators. We finished this parallel session. Don't forget to vote. You have a public vote, so there will be a winner of the public prize from your votes. You can vote on the platform, and there will be a prize for us. You can vote for your favorite your favorite poster, favorite oral presentation, and favorite video. So don't forget this. And you can now go to the hall of the posters to discuss with the others.
C'est pas mal, c'est dans le comité adulte.
Ouais, c'est ça, senior et tout.
Ah, ils avaient ça comme comité senior? Ouais, c'est bien des petits. Je savais pas que.
Bah, pareil, je savais pas non plus. C'est super. Ça va? Oui, oui.